Hello, everyone, and welcome to our class on connecting to R-viz, our Fiji R-viz in our case, as well as RStudio would be very similar to this. I'll point out instances where you would do something a little different on your local computer. Okay, so let's start a session. The website for this is in the syllabus, and you will have to have a VPN. Instructions for that are also in the syllabus. So if you haven't done that yet, you'll need to connect to your VPN to Colorado, or if you're doing this locally at home, you don't have to do anything. So this is what we have here. This is RStudio, or in the R-viz version of it. They're identical in many ways. So here's some of the basics that we'll go over. Starting here in this console here, where we want to go to the file path. Here, this is also in the syllabus. So the first button we'll learn is files, like where are you, where we were on the terminal. This is a very similar thing, but a much more user-friendly interface. If you click here, you can paste in the file path to the student class directory. Or if you're doing this at home, you're going to want to put the file path to where you want to clone the class repository. There's another video on the YouTube channel on how to get clone this repository and connect and collaborate on it. And if you're doing that locally, you'd want to just go to the local folder where you're gonna clone the Git site. Okay, so I'm already there. I'll cancel out of that, but I'll go to where you are now, students, here. And this is where you're going to want to do what I was just mentioning, clone the Git repository for class 2021, and do what we did in the previous overview of how to connect to Git, do all those same steps, and then you will end up with a folder that looks like this. But there's some other things we should look at here in this console. You can think of this as your sort of navigational window. There's also plots. Oops, I already have one plotted here. And this is a handy way to show you that you can export this and save it as a PDF or a file. And when you're done working, you can use this broom. Is going to be handy in each of these panels. It'll just clear all that up, and there's no more plots there. Packages, this will be important at the end of this lecture to install all the packages. We'll get to that in a second. You can do this here, or you can do it on the terminal. We'll talk about that in a second. But here's all the packages I have installed. And you can go to install and just type in the name of your package you want to install and install it. In our Fiji, we're going to always put it to here. However, or where our working directory is. And at home, you would do this where your R package has been installed. Okay, so I'm not going to install anything, but we'll do that at the end of this video. There's help here. This is very similar to the manual function in Bash. You'll see the Bash prompt is different. This means it's an R prompt with this arrow. And instead of man for manual, you do a question mark, and the function you're interested in. R will kind of know its base functions and pop these up. Again, this is why working in RStudio is kind of handy. So we want to do mutate and see what that is, and it'll populate to here. And what's interesting is mutate is being used by several different packages in R. We'll talk more about this as we go. This is just basic overview. But you can see that ggpubr is using it, dplyr and rstatics. Doesn't matter what those are for now. Dplyr is something we'll talk about more as we go along, but it's more of a base function. So let's click to see what that is. So unlike in Bash, where there's only one manual, in R there can be different ones. And it's good to be aware of which program might be running the same functions differently. And they'll give you some information about how to use it, what it does, same as the manual function. And then the viewer. So let's go back to files here. And once you've gone to the students folder and clone the Git repository, you'll see a Git connection here. And what we can do is start a new project here. So here you can create a new project, and we'll do that and let's do that now. Okay, so I'm not gonna save anything I've done here because we haven't done anything. And now we're gonna start a new project in a new directory, a new project, and the directory is where we just were, shares. I'm actually going to paste it. We made a symlink for this before, and you can use that to make this quicker. But for now, I'm just going to do this. Okay, so you want to create it in the students folder, not data. We'll make that folder in a second. So just go to the file path that you're on and hit create project. I have to switch to it right now. Okay, so cool. Now we can see there's an .r project, and you'll see your .git and your .gitignore will all be in here by now. And then another important button to talk about is, these are kind of obvious, but in here, we want to set our working directory. This is really important because this is sort of where R will use its base path file to go look for files or functions or other things. So it's good to always check your working directory. So right now, if I go to working directory, it's not here. So I'm going to set this as my working directory. You can see the R terminal did that for me. And so now if I went somewhere else, like here, I could just hit this button again and go to working directory, and I'm right back. So it's a nice way to navigate elsewhere and then come right back to where you're actually working. Set working directory and go to working directory. So set your working directory to here. This is where we'll be working, and at home, wherever your file path of choosing is. Okay, so then now here is the basic terminal. We'll go into this at the beginning of the next video where we actually start coding in R and analyzing data. But we'll be using this, but we'll also be doing this from a file. So it's good to know you always have a direct connection to R, and you can always talk to it directly when you see this type of prompt. But let's make a new file. I'm actually going to go back to my working directory quick here, and let's open today's actual file. You'll have this if you've cloned the repository. So don't worry about all of this, but basically you're going to see the title and some basic stuff that's separated by these. And this is the important part before we start the exercise, is to install all these packages. Each of these packages has different functions. You can use that question mark to ask about them, and we'll go over them in much more detail, but we'll need them for the lesson to go through this. So you can do that with install.packages. So there's instructions on how to do this here, where you'll go to install.packages, and then you just put the name in there that you see up here. And you'll install those locally, or you can do it as we mentioned here in install. Once you have all of these installed, and don't worry about this for now, we'll get into this as we go through the lesson, but in R you'll often give it a source path or function that you've made. Sometimes you make some sort of function and you want to use it a lot, and so you can tell R where that function is when you call it. Okay, so for now, go ahead and install all of those packages above, clone the Git repository, make a directory called data, and then on the website, all the data you need to put in that data is there on a link. Those will be the data files we'll be navigating and analyzing in the next class. So, and then just install the source functions as well. So for now, let's do that so we can get to actually starting to analyze read-in files the same way we sort of did in Bash previously, but now with R, go over some of the very basics of R code and how powerful it actually is in plotting data. And we'll actually go over a data output file from the last course where it says for 195 different proteins, how many sites they had in the genome. So we can look at how many times, who had the most binding sites and who had the fewest, et cetera. And we'll go through some of that. Before we go, just to finish the RStudio thing, we're now looking at things here in a document, and we can run this code. This is going to have an error. Actually, it might not. And the way you run it is by highlighting it and doing Control-Enter. And let's see here. Control-Enter, so it can find the file. I figured that would happen. So that's all good for now, but that's how you would actually run code, and we'll go over that more. But you'll need to do that Control-Enter, highlight it, Control-Enter for installing the packages if you do it on the terminal. And it's all written in here. And once you're done with a document like this that we'll go over later, just want to show you two last buttons here. Is when we're running this code, R will default to plotting it in this same window. And so we'll do chunk to output in console so that the plots and everything will come up in this panel under plots here. Otherwise, it gets kind of cumbersome moving through figures, and errors will also pop out there. Basically, everything that was going to pop out will pop out in this terminal, but you can switch that with this little settings button to chunk output in console. And then knit is going to be great because the way we're going to do these exercises and future ones is with this type of file, this R markdown file, we'll format it in certain ways that we'll go over. And then when we're done, what we'll get is something like this. After we knit it, it'll take all the code and sort of run it and put the output. This is Michael's markdown file from last year's class. And so you can see his code here, what he's doing, thinking about the plots that this code will make. So basically, in terms of reproducibility, you could cut and paste this into your terminal and reproduce this figure. So that's the advantage of working in R Markdown, where in each of these tick marks, with these 3 tick marks, we'll have what's called a chunk. And in that chunk is a set of code for a certain purpose. And then you can make a new chunk there. And for now, that's about all the buttons to see. We talked about knit, and we'll go over the environment once we start running things. The outputs from these will populate here in our variables, and we can inspect them here, and we'll do a lot more of that as we keep moving. And the nice handy thing is if we did change something here, save the file, we can get this to GitHub right away through here by clicking it and doing what we discussed in the Git video, committing it, and type to my collaborators what I did. I didn't do really anything. And then the classic push. And the nice thing is that you can do this all right from RStudio. And so it makes the Git life a little more graphical interface wise. So for now, all the data is at this link. All the instructions are shown here on what to do first. And then once you have all this installed and you have a directory that looks like this, where in data you have some of these files, you might not have all of them, but the ones we need for today's class are in that link on the syllabus. So once you have something like this, go to the next video and we will start navigating genomic data in R. All right, until then, code light.